afternoon, everyone. So welcome to the post-break session. Now we'll be starting with lecture second of the day, that is consolidated bioprocessing 2. So I invite Dr. Harun to kindly take over the speech and start the session.
Let's continue our discussion about the consolidated bioprocessing. And in this lecture 4, we are going to deal with the fungal CBP microbes, especially the filamentous and the white rock fungi. And we will finally compare the different CBP performance between bacteria and fungi. I read a lot of books about microbiology and yeast. The supremacy yeast is usually characterized as fungi. They have a lot of commons, including the metabolic pathway. Fungi and yeast share the same EMP pathway. EMP pathway from glucose through glycolysis and finally through the PDC inhibition to the ethanol. And another pathway is depending on the pH enzyme to the carbon dioxide. Theoretically, 1 gram of glucose can be converted into 0.51 gram of ethanol and 0.49 gram of carbon dioxide. It is theoretical data. Okay. In this list, I summarized some common fungi just for CBP purpose. From this slide, it details the glucose from substrate to the conversion. This mushroom is has the best conversion rate. It's 0.49, almost totally converted. Cellulose glucose almost totally converted, and the cellulose biomass theoretical. This also they use one straw to convert cellulose for ethanol. They get a very high yield, about 57.4%. The best one I would report is this one. This one is cross-up. Cross-up uses pure cellulose. It converts over 90% conversion from cellulose to ethanol. We also have some other fungi. There are no data from the cellulose direct fermentation cellulose. The specific fungi reach a comparable level to the yeast when we compare the ethanol production ability. However, for this fungi, the ethanol tolerance is usually much lower than the yeast. The ethanol tolerance is now still a big problem. Let's see the advantages of using fungi for the direct fermentation of cellulose. The fungi can be directly inoculated onto the lignocellulosic virus because it produces spores, a lot of spores. The result need of strictly anaerobic conditions. So we can use the normal fermenter. Sometimes we have to supplement it with oxygen. And another advantage is separation of fungal biomass is relatively simple because it has fluidity. From fermentation process, relatively feasible because it's biomass. The biomass of fungi is much larger in size than the bacteria. And then inoculation with conidial spores at high number levels. So its spores accumulate during the growth of at the last stage of the embryos. The number is of very high level. Saccharomyces is sometimes also considered as a unicellular fungus. These fungi are all cellulose-producing fungi, and this fungi we can find them in the market. This one we can purchase it from the industry. It's a trichodermal species. Trichodermal species is very common. It produces cellulose including EG, CBH, and the BGL. This is the spore. The spores are separated from the culture.
So this is marketed thing?
Yes, marketed. Produce the spores requires several days, but it's not a big problem because we use a solid state fermentation or semi-solid state fermentation. Solid state fermentation is not depend on the use of enzyme. It's depend on the use of straw, use of bagasse straw or other biomass materials. And this one is aspergillus. Aspergillus is in China is very safe. It's very commonly used fungi to produce a lot of chemicals. This one is penicillin. Penicillin is sometimes used to produce penicillin for the antibiotic agent. I isolated many penicillin species from the rotted orange. From the rotted orange, there are abundant glucose oxidase. It is used for the refinery of to produce glucose or gluconic acid. And this one is a plant pathogen. Plant pathogen. It causes heavy diseases to the level of plants. Its name is Fusarium. Let's see the trichoderma first. The trichoderma cellulose has a very high CBH content, especially trichoderma ricci, especially the cellulose divided by multiple times. The CBH content is very high. It is important for the complete digestion of biomass cellulose. The strain A10 could grow only under aerobic conditions, but it could ferment cellulose and the sugars under anaerobic conditions. This strain is special. It can be used to do fermentation under anaerobic conditions. It accumulates 5 grams per liter ethanol. It is a very low level. It means its ethanol tolerance is poor. There are some engineering efforts on this strain, the trichoderma species. Someone they do the overexpression of cellulose proteins and it could be used to enhance the ethanol resistance. So one to the genome sampling of TVC to produce ethanol under anaerobic conditions to get a final glucose conversion rate 0.21. 5-fold of the wire. The engineering work improves the final results, CBP performance of it. Next, filamentous fungi is Aspergillus. Aspergillus is in biofamilies. The most important role is to produce a BGL, and its activity depends on the strain. Different strain produces different products. The author scored this grain a lot of characterized Aspergillus species for the fermentation of cellulose to ethanol. We found that all of these species can be used as CBP microbes. The fermentation of corn spore, including the corn stalk and the corn cob, can be fermented by Aspergillus oryzae. It yielded ethanol to 0.43 gram per liter per hour. It's a very good productivity. With maximum concentration of 20 gram per liter. It's 2%, 2.5% in volume. The engineering of Aspergillus mainly focus on expressing some new genes from other skin in order to promote the ethanol concentration. But the ethanol tolerance is still a problem, unresolved yet. There are many new work they focus on this to solve this problem, but no good results until now. This fungi is the third one. It's Fusarium. Fusarium is very common. You can easily get it from a banana tree like this. This tree was caused by this bacteria, this fungi. It is not healthy. It is not healthy, and it is now visible. Visible means it cannot live for a long time. It cannot produce banana yet. And this one is a tomato. All of the leaves were rotted due to the growth of this fungi. And this is rice. The whole straw of rice is heavily rotted due to this disease. So if we use this stem, there is a risk. There is a heavy risk because it may cause plant disease and cause a lot of loss to agriculture. So in my lab, Dr. Liu Ming from Guangdong Agricultural Science Institute and we worked together to study the antimicrobial peptides. We tried to find a peptide that can control the disease caused by the Fusarium. And we tested the application of peptide like this. This is the sequence we designed it by ourselves. This is the model we simulated using a software. It has 23 amino acids. And the molecular weight is 4,000. The PI is 9.34. This is the net chart, and we tested. We predicted its hydrophobicity and hydrophobicity. We produced this antimicrobial peptide very pure. The HPLC results indicate that its purity is over 99%. It's a food grade in China, food grade. So we also try to use it for human health, not only for the agricultural purpose. Nowadays, the peptides can be directly synthesized using biochemical way. But the most feasible way is to synthesize a gene and express it to the nuclei, and the nuclei produce it more efficiently. Because the biochemical synthesize, sometimes the sequence is not correct. There might be some errors in the sequence. In my lab, my students, including Dr. Puneet, they helped me a lot to test the antimicrobial activity of this peptide. They studied a lot, including the fusarium. To prevent the growth of penicillium, fusarium, the relative dosage of this peptide should be very high, 200 ppm. But for bacillus, 25 ppm is enough to prevent the growth of all the bacillus cells grow on the agar, on the nutrient agar. So this work, this table, this result from this table, spends us a lot of time, because we need to do the dilution, we need to do the coating, and we need to cultivate it in the incubator, and finally we have to count the number. It costs us a lot of time. So what's our method to test the antimicrobial activity? Do you have a better method?
Is it the turbidometric method or the weight?
Micro-dilution broth method, MIC.
MIC.
Or in highly zigzag legs.
Sometimes I use a GC. I use GC to, for example, grow a bacillus in a flask, and it will produce some products. I will test the products, for example, but it does not work for all of them. The most successful one is the nuclei. Our lab published papers based on nuclei because there are carbon dioxide and change, and we can test the products value. But it's not a good way. I think the plate, using the plate, it's better. The data is very stable.
This dosage is mixed in the media previously, and then we inoculate the cultures for it.
Yes. Yes. Because all of these bacillus cells separate into pores, into liquid, and to a certain number. For example, 8 or 9. Then we add the peptides into the liquid, and this dosage. This liter. Liter means the volume of the liquid totally, not based on the number of the cells. Yes.
So what is the relationship of studying this antimicrobial study with the CBP?
With CBP, yes, it's like this. If we want to use CBP, there is a risk because it will cause disease. And if we have this antimicrobial peptide, it may help to control the risk. Yeah. And for the fusarium, for this fungi, the enzyme production secretes cellulose, including EG, CDH, and the GIAN, and the hemicellulose at a very high level, anaerobically, and it works anaerobically. It also ferments monosaccharides hydrolyzed by the biomass to ethanol under an aerobic or micro aerobic conditions. The ethanol tolerance is higher than aspergillus. So you can give it the final ethanol concentration can reach 72 grams per liter. And it's one of the best results from a non-yeast microbe, 72. And the sugar uptake, the glucose is important to the cell and enters glycolysis pathway. And we showed it in the first picture. Hemicellulosic sugars, including mannose, galactose, and ribinose, enter an alternative pathways to generate glycogen and intermediates, and finally enter the glycolysis to bioethanol. This pathway has been characterized. So the stability performance, the direct fermentation of cellulose resulted in an ethanol concentration of 9.6 and 40.5. It is a very old water, and it requires 6 days. And compared with the yeast, it grows very slow and consequently requires for a long time cultivation. Right? The last one is penicillium. Penicillium is usually used to produce cellulose. Very little work about using penicillium to produce ethanol. I found only one work. It's very old. And ethanol is included in the volatile products. Not only ethanol, but also others. Okay, the next one is white rock fungi. White rock fungi is can digest the lignin, can produce a lot of different oxidase, including lignin peroxidase. Lignin peroxidase is very important for the degradation of non-phenolic lignin unit. We know that lignin has a phenolic part, and a non-phenolic part. And a phenolic part is here the ring can be more readily broken down. But if they form ether, carbon, oxygen, carbon covalent bond, it will be very difficult. It will be very difficult. But this enzyme is good. This enzyme can directly degrade the non-phenolic lignin units. Not only this one, but also this one. This one is newly identified as volatile versatile peroxidase. It also can also be produced by white rock fungi. They are different because when we are going to use this LIP, there must be a co-substrate. Co-substrate means there must be a very small molecule, similar to a lignin unit. Okay. For example, a phenol derivatives. If there is no co-substrate, the efficiency of this peroxidase will be very low. And for versatile peroxidase, it works much like the HRP. It requires only the hydrogen peroxide. And the white rock fungi also produces other hydrolysis oxidase, including lactase. Lactase can also oxidize the phenolic part of the lignin. I will introduce it in detail this afternoon. And another one is manganese oxidase. It is depends on the presence of manganese metal ion here. This picture is white rock fungi are isolated from a mountain in Guangzhou city. This HLS is the name of the mountain. I cut it off and try to do the dilution and get a purified slate. Many research papers they reported using vital fungi for biological treatment of biomass because they can be used to delignify. After the delignification, the cellulose content can be largely enriched. It also saves the chemicals, the use of chemicals. However, this process is quite long, usually for 20 days, 30 days, or several months. It's a long time. For example, if we use this mushroom, we cultivate it, grow it in our lab, use a plastic bag, this is the plastic bag. Okay. In the substrate, the major component is rice straw. Rice straw. Okay. And the oxidase are secreted by the methane in the residues in the substrate. This is the fruit body. We can also test the enzyme activity in the fruit body, but the level is not very high, very low. So a major application of white rod fungi in bioethanol production is to delignify the biomass. It saves energy, saves chemicals, but consumes a long time. Then in one of my projects, I use cultivation of mushrooms or residues because it's full of cellulose, full of biomass, and this biomass, this residues, has been lignified, has been enriched with cellulose by the mushroom. So it is more readily to be digested by the enzyme. I tried 3 different types. The first one, one round. One round means the mushroom fruit body grows for one time. Second round means after cultivated, harvested, it grows again. Okay, after maybe 9 days, 10 days, it will produce fruit body again. Then the lignin in the residues will be further lowered. And the third round, this mushroom can produce fruit body for 3 rounds. Then the results suggest that the third round, the residues is much more ready to be digested by celluloses. Here I listed the performance of the white rod fungi as the CBP microbes. Use this fungi, use this mushroom under anaerobic conditions, the ethanol can be produced. Ethanol is not very high. The content is 3 grams per liter. It's not very high. At the end of this lecture, I compared the performance of different CBP microbes from the ethanol yield and the ethanol tolerance. We can take a look at this figure. Over 100, around 100 is supremacy yeast. Supremacy yeast can tolerate a very high content of cellulose, and the conversion rate is usually around 90%. This one, the Pronsa is the ever reported, the best one ever reported. It can convert biomass cellulose to a higher conversion rate and accumulate the ethanol titer to 2 or 3%. Here are my 2 questions. One question is it safe to use fusarium? Fusarium because it is a plant pathogen. Is it safe to use this one in biorefinery? I think it's a big problem. So that's why we developed the microbiome peptides. So can we use it? Let's say we use it.
The peptide, gene of the peptide is incorporated in the fungi.
Yeah, we read a lot of publications, and we try to use the computer, use the database to design the sequence. The peptide sequence is not existing in nature. It's a rational design. So what can we do to reduce this? For example, it's now it is harmful. If we can do something, if we can take actions.
We can perform the gene sense.
Yes.
Yes.
Designs.
Okay.
Okay, to engineer this fungi, right? Second question is try to design an experiment for screening Pacinium species with CBP capsule because no one do that. No one do that. I do not find a paper to use this strain for CBP purpose. How can we design this experiment? I'm asking my student to do this work. And do you have a question about China, about Guangzhou? In my city, the most famous architecture is Guangzhou Tower. Guangzhou Tower is one of the tallest buildings in the world. It is made of steel. Only at on the top there is a dining room, and it's not easy to get to the top of it. You need to take 2 or 3 lifts to change. I lead Professor Shula experiment to visit there. They take pictures, but not out went to the top. And I also showed him the Chinese high-speed railway. We take the first train from Guangzhou to Shenzhen. Shenzhen is a nearby city. The train is good, but he read on the screen in the car. He told me that it's not 300, it's 200. Next time I didn't let him to another city to Shanghai. The train is newly developed, very new. It runs since this January. Yes, and the speed is 350. It's heavy. It was heavy.
We are 70 or 80 kilometers maximum.
100.
100 maximum.
So I welcome you to take a look at China, visit China. From the police, I led him to my in-law, visit him. My grandmother, my grandfather's family, his house. We have a dinner. We had a dinner together. But in my hometown, we use a lot of meat and a lot of fish. When the police add only vegetables.
So for those who don't know, Puneet was a PhD student. So after completion of his PhD, he joined this app. So Puneet is our own student who is working with him.
Nowadays, it's not easy to get a visa to China for 3 months. For short-term visit, for example, 1 month, 2 months is okay. For long term, it's not easy. For example, if you are applying for a working visa for 2 years, 1 year, 2 years, they need admission. There's a license number. It will be very easy to write a invitation letter from the company. From the company, you will get a visa more easily. How long? 5 hours from Newry to Guangzhou. 5 hours.
So anything to be discussed on this topic?
There is a detailed lecture on microfungi next session.
Next session.
Detailed lecture on microfungi.
Yes.
Shall we discuss in this session? Or next
Afternoon.
afternoon.
Before it, yes. I will introduce my research work, my data about this, many other unions. All my research work focuses on union.
It's over. We have some more lectures. It's over.
This lecture is over.
What is it?
Yes.
Okay, so open for discussion.
So,
Yes.
beside using physical for the discussion production, why we don't go for the engineering of ethanol tolerance for cytolysis? Increase the ethanol tolerance for cytolysis.
It's like this. In China, we have wine. We have wine of very high ethanol content. And from the wine factories, we can get the ethanol tolerant yeast. It's not a big problem. I introduced to you the MDU. MDU, this company provides ethanol that tolerates to high sugar content, high ethanol content. The yeast is very good.
That can be using this bioproduction.
Yes.
So, besides using physical, we can go for that and that with different layers.
We got the. You were asking about scale development, I guess.
In this, I can make some niche how to do.
Dr. Hall, what you were asking is, can we make a recombinant yeast which can sustain high level of sugar or high level of ethanol tolerance? Can we do some gene engineering for that? Putting some gene so that it get resistance to high level of ethanol.
We try to use the evolution method.
Because the yeast that is present in white and grape factory, they are not producing all the enzymes that are required for the liquid phosphatases. they are not producing that. So that's why we can't use. Maybe we can.
No, see, here, there are things where one is valuable and one is exposed permanently is. So what Saccharomyces can do is it can only convert the sugar which is available into ethanol. It doesn't have a machinery to degrade lignin or cellulose. For that you need ascorbic filter. So there is two separate process. First, you need to convert your biomass into sugar. That is called the deconstruction of biomass. Deconstruction of biomass into sugar, and then that sugar can be hexose or pentoses, either 5 carbon or 6 carbon. If it is 5 carbon sugar, you need a picture of some yeast which can convert, which have the pathway to convert 5 carbon sugar into ethanol. Or you could go for Saccharomyces, which has the ability to convert 6 carbon into ethanol. But Saccharomyces doesn't have a pathway for pentose. Like my the picture doesn't have a pathway for hexose. So what can be done is what people have done. They have designed a pathway. They have inserted a pathway from Pikia to Saccharomyces because now Saccharomyces can have both the potential. It can convert both hexose and pentose. In that way, what you can do is after saccharification or after deconstruction of the material, now you take both the sugars. So if you are using bicycle Saccharomyces, only hexose will be utilized and pentose will be left. Again, you have to use another yeast for that. You have to separate pentose and you have to use other yeast for fermentation or some other process. You have to use for some other strategy. But here, if pathway is engineered into Saccharomyces, and you can use that, it can be more convenient, efficient.
In the same way, we can add genes for scarification as well in Saccharomyces.
Yeah. But see, there is a limitation for that. Our microorganisms has to be controlled, regulated. Gene has to be regulated. Which gene you want to express first, and then there has to be some cascading of it. So there is a method called gene targeting or gene cascading, where you can put more than one gene in a cassette. So it's not so easy to see people for research purposes, but I have seen for research purposes people have devoted multiple genes in one cassette. But practically doesn't work. If you want to access more than 2 or 3 genes, it is very challenging because energy is limited and you have to, and the pathway is limited. You have to both sacrifice the perimeter and then you have to submit also. So you have to control detail. Naturally, nature has not evolved like this. It has given the potential, segregated the potential in ascorbic fungi and ascorbic yeast, which is Saccharomyces. So it's not that efficient.
If we insert only one gene also, then what is the success rate?
So it's working going
We have to do suppression also,
It
reduce
is lost.
the efficiency.
It's lost.
Yeah.
It is lost. For that, they give our research cost.
That is okay, but we have a opinion what do I think? Instead of going for genetic engineering, can't we go for evolutionary engineering?
That's why this year the got to direct evolution. Whenever direct evolution is given some electron PCR and you evolve like you have done so people have they have done artificially they have evolved genome also and the pathway also. That can also work. Even I have seen white even PhD also and recently lot of cases we have seen that when biotech culture will lose producing, everybody are producing that. Sometimes we see that they don't produce and they don't convert sugar into ethanol. Biotech also very problem comes and they reject also. Sometimes I'm saying by subculturing, unfortunately, not chocolate, subculturing private fungi or because it may be in subculture 100 times in 5 years, I don't know how many times it touches. And it will be subculture. It loses its potential to produce the enzyme what it do. If it lost the potential at fourth year or fifth year of the basic. That is very tough task. And then what we have seen that we have transferred that fungi into the natural system, like we have transferred into linear rich substrate again. Operated in the linear rich substrate. Yeah. And after a few months of growth, it has regained potential because the gene has not lost. The only thing is that forget to produce that enzyme because you are already in a media which is not a natural choice for a fungi. So we think that not extract or PDA is not natural. So in biomass also happens, but see all it's very like you see they have different strain and they don't release the strain, they are recombinant strain. And they keep the stock and if they lose the strain as you were telling that they will really you won't get the same taste and same fear of buying their own. So they have a mechanism to preserve the gene. Yeah.
Fourth lecture and ninth lecture. What should we see microbes?
Your son.
After fourth, it's ninth.
Anyway, maybe that will be just
Maybe Professor Shukla's trying to
he's going to use monitor
So overall, the strategy which was discussed today, that was simultaneous fermentation, saccharifugation and fermentation. That is, in terms of efficiency and yield, that is far better than the separate saccharifugation and fermentation. So, I think nowadays the focus is shifting on simultaneous saccharifugation and fermentation. And we have to design such organisms which can carry out both the processes. So that's the new challenge which is coming up, and many of the scientists have got tremendous success in this strategy. So even we are just thinking of maneuvering the yeast, but it's it's not so easy. Maneuvering the wild type organism. But some development has been made in generating the first synthetic organism. That's old news now. And the first synthetic microorganism was generated in a lab, the great Mendel's lab. So he actually assembled all the genes which are required for fatty acid synthesis. So he is developing some microorganisms which will synthesize high amount of fatty acids. And later, we can convert these fatty acids by transesterification reaction to diesel. So what this lab is working towards is production of diesel in a fermenter. So the strategy is to assemble all the genes which are required for fatty acid biosynthesis. So he is assembling genes from C6 to C12 carbon chain length, and what he plans that these microorganisms will be secreting fatty acids, and these fatty acids can be used as fuel by a simple reaction that is called transesterification, and fuel can be made in fermentation by fermentation. So people are very seriously working on engineering the pathways so as to come with some good results in terms of fuels. So metabolic engineering is something which is really coming up, and what we are doing in labs is still basic research, but people have advanced to metabolic engineering.
When 15-20 years earlier, some Japanese or Chinese people they had pathway both fatty and exposed both pathway into chloride. So they had one channel that they had codon modified, they had codon modification because they can't express fungal gene into bacteria and vice versa. So they had modified the codon, fungal codon, which was for different pathways of exosomes and those fermentation into the E. coli. And they are expressed both of them because fermenting with E. coli is much better easier than with isolated fungi or anything. So, and you know that E. coli is a model organism, because 15 years back, they were having a whole genome of E. coli, but not of these yeast. So people use E. coli as a model. So they have done both pathway in bacteria, and they have produced ethanol in a very efficient way. Nothing about people are doing that. And I think the smart way of working in a lab, where you have some constraint resource opportunities, people who have already established a method, you can collaborate with them. Because a single lab can't do each and every optimization experiment. So if someone like he has not produced enzyme, how do you have not produced enzyme? He purchased enzyme cellulase, best fit cellulase, and he has used for his application and fermentation and also yeast. And he has designed a process for technology, how to whole engineering part. He has taken care of whole engineering part, and all basic things. So if you want to make a practical and commercially viable system, once you look into different labs, a lab which is well efficient and well established and patented, a lab having a technique how to do downstream processing of ethanol, because that is also a big challenge. You have ethanol in a lot, you need to separate. There is no use of having good yield, because you can also lose that ethanol which you are having in a lot during downstream processing. So what is the you are saving a lot, but a lot of getting in downstream, and you are losing ethanol. 5% loss of ethanol during downstream. There is no use of ethanol getting you lost the gain whatever you are supposed to have. So better to go for all the techniques, all the people who have done some good work. First challenge is your treatment problem. First is your treatment substrate, because we all obviously we go for some legumes substrate. We go for legumes like a country like India and China, there are a lot of available series. So that is the first challenge to remove your lignin rich substrate, lignin, at least to partially remove the lignin, so that your stainless and metallus enzyme to enter into the substrate. That is making a gap. We have seen a figure where he has shown that there is a gap and cellulose enters to the gap. So making the gap is very important. What I feel that instead of having a complete treatment, you do some partial treatment with the microbe fungi. Microbe fungi is to break the venous resource. So it will make some gaps so that those other ascomycin fungi or cellulases or enzyme, whatever process you follow, CBD or it will work better. So it's not that you completely remove the lignin. Using biological that is because lignin is weak acid. You can't remove lignin completely. You could remove partially because it is not to capping or blocking cellulose and hemicellulose, so that it protects that cellulose and hemicellulose with foreign paste or something like that. Protects the root, and it acts as a binding material. So you make a gap. Slight modification. So that's why we have shown the Apex method,
Yes.
explosion method. So there is something like CADEX method also, chemical method, CADEX method, we use the CADEX. So a lot of methods people have used just to partially break. A lot of chemical method, a lot of physical method using like steam explosion method, where high steam, high pressure, ammonia.
Treatment, heat treatment. Still, it's like the pre-treatment is a very important step. And, a lot of time goes in pre energy also. Especially, I told you about water also. And especially when you treat it with acid, if you treat it with acid or alkali, then a lot of efforts go into neutralizing the iron hydrolyzed.
Why don't you use water for that?
So water is
And
again.
then you use acid to see, using mineral acid is very economic, very cheap, very economic, using HCl or H2S, it's very economic, rather than using enzyme. You can use enzyme, you can use acid also. But another challenge is when you use acid, you get, obviously you get the sugar. If you use alkali, you can remove bleeding also, you can sodium chloride or sodium sulfide. You can easily remove bleeding, you can get cellulose. But when you use acid or alkali, for example, you use acid for aggregation, you will get a lot of acid derivatives, sugar derivatives, like fulans and sulfuric. And fulans and sulfuric, again, they are inhibited to microorganisms. Yeah, in the yeast. So either you have a yeast which survive at high level of fulans, or you do some apply some method to remove detoxification, remove the fluorescent. Again, you just sublining or some other charcoal method, sublining method. Then at every step, there may be some loss of sugar. Each step there is loss of sugar, loss of energy. So that is also very important because I think we will use 5 to 10% sugar.
Each step is lost. People are also working towards producing low lignin plants. They are engineering the pathway so that the plant has less of lignin. But the problem is that the plant doesn't stand. The lignin content is reduced. So this is the skeleton of the plant. So the plant falls. They've been certain success, and in fact, the plant is not able to survive if the lignin content is low.
Have you heard about what the radical acid? Yes, not. That's what no? People are using RNAi technology, RNAi technology, to make to manipulate the pathway, phenylalanine pathway. Pathway or phenylalanine pathway is a pathway for the synthesis of lignin, where you make lignin monoleic phenols, polyphenol, sulfonyl, and formyl alcohol. These are all 3 monophenols which combine in the ratio to make either hardwood or softwood. So these there is one pathway like formylic, a lot of acids are made, formylic acid, citric acid. So these are all in Pal pathway, phenyl pathway, starting from amino acid phenylalanine. So what people have done, there are so many steps in pathway. They have chosen steps and tried to down-regulate the gene. They have tried to down-regulate the gene, and they did tissue culture because it plant has people there along with the background. It's already background. So they have tried to make a plant with lesser lignin, more of cellulose, lesser lignin. That is fine, but the lignin has other role also. Those plants will not survive the natural phase. Lot of disease lot of disease can occur, and it has to stand also, it has to grow also. People are they have better plant in sugar. They have calculated, they have estimated the lignin is less, but they were not able to grow in vine. So you can summarize. Better how you can summarize. You want to say something?
So with this, we come to the end of this technical session. Dr. How you want to summarize the session? Please.
Thank you for your attention. This episode, I will invest in the tutorial we will enlarge consolidated file processing concept. We will talk about first the CBP of lignin. It's a new technology. Another another content for this afternoon's tutorial, well, I'm going to share my results about the lattice with you. Lattice, all of this research done for for the panel with the help of Dr. Neil Sun. We have published most of the results. Yes. Thank you. Thank you for your attention.
Now all will disperse for lunch and we'll meet here again at 2. So I request all the participants to come back at the seminar room at 2. And then we'll be having tutorial for 2 hours.